Hello, everyone, and welcome to today's live broadcast, Fast Glycan Labeling and Analysis, presented by Andras Guttman, PhD, Senior Manager of Applications for SCIEX Separation. Before we start the presentation, I want to advise that poll questions will appear on your screen. You may select your answers, then close the poll questions by clicking the X in the right corner. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by SCIEX. SCIEX helps to improve the world we live in by enabling scientists and laboratory analysts to find answers to the complex analytical challenges they face. To learn more about SCIEX, please visit SCIEX.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or you can use the Q&A button to let us know you're having a problem. Thank you. Now I'd like to introduce Dr. Guttman, recipient of the 2016 Bioprocess International Best Technology Application Analytical Award for his work on SCIEX Fast Glycan technology. Andras Guttman currently leads the application efforts at SCIEX Separation, Brea, California, and holds the Translational Glycomics Professorship at the Horváth Csaba Laboratory of Bioseparation Sciences at the University of Debrecen, Hungary. His work focuses on capillary electrophoresis and CEMS-based proteomics and glycomics analysis of biopharmaceutical, biomedical, and cell biology interests. Dr. Guttman previously held academic appointments at Northeastern University, Boston, Massachusetts, and a Marie Curie chair at the University of Innsbruck, Austria, as well as industrial positions at Novartis, La Jolla, California, Genetic Biosystems, San Diego, California, and Beckman Instruments, Silicon, California, developing high-resolution capillary electrophoresis and microfluidics-based separation methods. Professor Guttman has more than 269 scientific publications, wrote 35 book chapters, edited 4 textbooks, and holds 19 patents. Until recently, he served on the CAS board and as president of the Hungarian chapter of the American Chemical Society. He is on the editorial boards of numerous international scientific journals. Dr. Guttman graduated from the University of Veszprém, Hungary in chemical engineering, where he also received his doctoral degree. He was recognized by the Analytical Chemistry Award of the Hungarian Chemical Society in 2000, elected as a member of the Hungarian Academy of Sciences in 2004, named as a Fulbright Scholar in 2012, received the CAS CE Farm Award in 2013, the Arany János Medal of the Hungarian Academy of Sciences, the Proscientia Award of the University, and the Denis Gabor Award of the Novafor Foundation in 2014. Dr. Guttman was also the recipient of the 2017 Dal Nogar Award at the Delaware Valley Chromatography Forum, which he received at PitCon earlier this year. Please join me in welcoming Dr. Guttman. I will now turn the presentation over to him.
Thank you very much, and I really appreciate the nice introduction. And I'm very excited that I have this opportunity to talk about the fast glycan labeling and analysis kit that we have been developing at SCIEX, and as a matter of fact, which won the Bioprocess International Award for Best Technology Application in Analytical Chemistry. So the overview of my publication, the overview of my presentation is that first I would like to talk about the importance of glycan analysis of biotherapeutics, and then shortly discussing capillary electrophoresis as an established and routine analytical platform that can be used for glycosylation analysis, and then the SCIEX new award-winning fast glycan labeling and analysis technology, and a summary. Why is it important to check the biopharmaceuticals these days? And as you see from this slide, out of the 10 highest sale drugs these days, 7 of them are large molecules, and 5 of them are monoclonal antibodies. Therefore, it's very important to analyze those monoclonal antibodies from the glycosylation point of view, as well as, of course, everything else. But the IgG1 molecules, which represent the majority of these monoclonal antibody therapeutics, has one N-linked conserved glycosylation site at the asparagine at 297, and the variability of this glycosylation is dependent on the host cell line and expression conditions. I will be talking about that one a little bit later. So there is a structural diversity of this glycosylation, meaning like fucosylation changes, galactosylation, sialic acids, and even monosylation. So this glycosylation itself at that conserved site determines the biological activity of the compound, the physical-chemical properties, and some functions, some very important functions, like ADCC or CDC functions, which are the antibody-dependent cellular cytotoxicity and the complement-dependent cytotoxicity functions. So the importance of checking these IgG glycosylation sites are at clone selection, obviously, to pick the right clone for development, product characterization during the process, and at lot release. So let's see first the significance of these glycosylation sites. So as I mentioned, for example, galactosylation is very, very important because it increases or even decreases, if necessary, the affinity of the Fc gamma R, and depending on the specificity of the pairing. On the other hand, the removal, for example, the fucosylation right here, that affects the ADCC function. So it's an exponential function. So if the ADCC function is the goal of the product action, then this fucosylation shouldn't be there. So we need very, very precise analytical methods to reveal if there is fucosylation or not. On the other hand, we can have the silylation part too, like here. And the silylation part of this glycosylation is responsible for the anti-inflammatory properties. And finally, like the glucnex in the middle of the molecule are very, very important for the monomer binding lectins. So these are mostly the individual contribution of the sugars at the structures. So during the manufacturing process of these recombinant therapeutic proteins and mostly like IgG molecules, during the clone selection, it's very important that we understand that the glycosylation is such what we would like to have at the end product. And the clone selection is like a large-scale, high-throughput methodology. Hundreds of samples should be analyzed in a very, very short period of time. But all over the process, on the manufacturing process, of course, this glycosylation should be characterized and even validated in some extent. At the end of it, with the QC part, the companies need very, very high resolution and very, very precise quantitative measuring tools to check the glycosylation of the product which should be released. So the glycosylation processing itself can have some problems, and many factors contribute to the alteration of this glycan processing during the recombinant glycoprotein manufacturing, such as the expression levels of the processing enzymes on the host cell line, the monosaccharide donor level, the cell signaling pathways, loss of cellular organelles which actually put the glycosylation together, the mutation of the gene silencing and overexpression of the glycosyl transferases or glycosidases, and of course the bioprocessing environment such as temperature, oxygen level, and so forth. So if we aim for the quality by design, then we have to understand the sugar-function relationship, for example, the core fucosylation for the ADCC function, what I mentioned earlier, and the selection of the cell line, which should be appropriate for the glycosylation machinery that puts on these glycans, which are necessary for the mode of action. The characterization approaches are really plentiful. And people have been using different methodologies because this IgG or any other therapeutic proteins should be very, very heavily analyzed. out of these methodologies and out of these structural changes that should be analyzed, we picked the glycosylation. It is what we would like to talk about in this webinar today, and mostly using capillary electrophoresis, also mentioning that the CSIMS or SCIEX's new technology to connect mass spectrometry with capillary electrophoresis in the single dynamic process will be mentioned as well, but mostly focusing on capillary electrophoresis. So what is the challenge here? The challenge is that carbohydrates are very complex and very diversified structures. There's no chromophore, no fluorophore groups for UV or fluorescent detection, and mostly not charged unless they are silylated or maybe sulfated or acetylated. So the analytical methods in hand, what we are using today, is of course capillary electrophoresis is a very, very high performance methodology. Liquid chromatography and its subtypes can be used, obviously. Hyphenated methods like CEMS, CSIMS, or LCMS, and the old-fashioned structural characterization options with mass spectrometry or NMR. Let's see how capillary electrophoresis works. Capillary electrophoresis separates species based on their charge to hydrodynamic volume ratio. And if you see this slide, the A and the B traces are the same glucose oligomers with 1,4 connection, but one of them is an alpha and one of them is a beta section, which means that the shape of the molecule is different. As you see, in trace A, it's with the alpha 1,4 linkage, it's a helical structure. With the beta 1,4 linkage, it's a linear structure. And just based on this shape difference, the individual oligomers of these oligo sugar molecules are migrating differently in capillary electrophoresis, proving the extremely high separation power of this methodology, which again is based on just the shape of the molecule. So, what are the challenges of the industry in that way? The time what is needed for the glycan identification. Imagine that you have a bioprocessing unit and you would like to check if everything is going well or not in that unit. You need to analyze your sample very, very quickly if you have to make changes in the pH, the temperature, the oxygen, or anything, what have you. So the rapid analysis is key. And it includes sample preparation, the length of the separation time to give you the right answer. The complexity of the analysis should be such that you can really use the Glycoinformatics tools for data analysis and identifying those key sugar structures what should be right there at the molecule. So to meet these analytical challenges with the processing time and resolution and automation, this is just an example what I would like to show of the separation of a standard mixture of glycans. And this shows the separation of the fucosylated and afucosylated major IgG glycans with the presence of a high monostructure molecule, the mannose 5. As one can see, within 4.2 minutes, all of these structures can be very well separated. And this is what we would like to show, how the new fast glycan labeling and analysis kit can fulfill this mission. So, starting with the release of the glycans, with the enzyme which mostly using PNGSF, the next step is the labeling of the molecule. As I mentioned earlier, unfortunately, these sugars don't have any fluorophores or any other UV absorbing groups. So in this case, we use the 8-aminopyrene 1,3,6-trisulfonic acid to do the labeling. And that also provides us a very, very heavy charge, like 3 sulfonate groups, on the molecule. And as one can see, it's a very simple shift-based formation, but will be reduced after the shift-based formation with sodium cyanoborohydrate or any other hydrogenation reagent to a stable conjugate, which can be analyzed later by capillary electrophoresis. It's a very, very simple and efficient reaction. It has much higher than 90% labeling efficiency. It's non-selective. It's uniformly labeling all the structures. it's very, very easy to quantify because we have only one fluorophore per sugar molecule, because it always reacts with the reducing end. So it's one fluorophore per sugar molecule, which means that if we have twice as high of the peak, then we have twice as much of that particular sugar. And it is very, very sensitive by detecting using usually used argon ion or solid state lasers with excitation of 488 nanometer and emission of 520. Again, the reaction can be done very, very rapidly with optimized parameters, but this R group is what represents actually the sugar structures. And of course, we have somehow represent these sugar structures in the rest of this talk. And since these would be the structure themselves, I don't think everybody would like to see those structures actually filling up these slides. So we should use some kind of a simplified methodology to show these sugar structures. And we are using the Oxford nomenclature for that one, which is shown right here. And the monosaccharide units and the linkage positions and the linkage type are decoded like shown here. And mostly we will be using just the abbreviated form of, for example, this structure here, the FAB2BG2S2, which means that it's like practically a biantennary structure with a bisecting glucan, a core-fucosylation, and 2 sialic acids. So let's see the heart of the technology. And this is a magnetic bead-based sample preparation technique with the highest importance of not necessitating any centrifugation or vacuum centrifugation steps at all. And that's one of the major and most important message, and that's the reason why we can get it done as fast as it's shown right here on this slide. So the first step is that we have the magnetic beads and the sample component in the reaction vial. After that one, we denature the sample at 60 degrees at 8 minutes. After denaturing, we add the PNGSF, and everything the PNGSF mediated removal of the glycans from the backbone of these proteins are done with the presence of the magnetic beads, which means that right after this step, if we add the appropriate amount of acetonitrile, like an organic kind of an agent, all the sugars will be bound to the magnetic beads, which actually let us pull down all of those glycan structures on the surface of those beads and get rid of the supernatant without doing any centrifugation or vacuum centrifugation steps. We are concentrating all of our sugar molecules on the surface of those magnetic beads, which allows us in the next step to do the labeling right at the surface of those beads. So we add the labeling solution, which washes off all the sugars from the surface of those beads, and the labeling reaction is done in 20 minutes at 60 degrees centigrade. One more important part of this one, that we are doing all of these individual steps in a way that it will be completely liquid handling robot friendly, which means if you would like to use a liquid handling robot, you cannot really cap these vials. And it's even better for us because in that case, if we are doing this labeling reaction at 60 degrees, during 20 minutes, it's a significant loss of fluid, so which means it's a natural concentration of the labeling reagents during the labeling reactions, which even helps us to speed up the labeling. So that's the reason why we can get it done in 20 minutes, which is much faster than the usually used overnight kind of labeling methodologies. After the labeling step, again, we have to clean it up. So we add the appropriate amount of organic, then all the APTS labeled glycans will be bound again to the surface of the beads. We can wash out the excess dye, and then finally elute the labeled sugars from the surface of the beads, but can directly go into the capillary electrophoresis unit with laser-induced fluorescence detection. So that is the heart of the kit itself, and that's the reason and the way as it can be done in 60 minutes. If we check again the individual steps, the first first minute is the magnetic bead preparation. The second minute is the glycoprotein sample addition. I would like to emphasize that here within this 60 minutes, we have every single step included. The pipetting, moving the vials from one position to the other position. It's not only just saying that, oh yeah, we have the digestion in 20 minutes and the labeling in 20 minutes, so it's 40 minutes. Not. It's not 40 minutes because everybody needs time to put from here to there, to pipetting and all of those things. This 60 minutes includes every single step which is necessary to do the full sample preparation, either manually or by a liquid handling robot. And then, after the sample preparation, we inject our sample in the capillary electrophoresis unit. We have a 20 centimeter separation distance, and as one can see, in like a little bit more than 4 minutes, we got full separation of all of those glycans which were released. This example is from a human IgG molecule that was practically prepared using this technology that I just shown. And all of those structures which are numbered have been identified and shown in this big table, which is a little bit too crowded, I understand, but that's the way to go if you have too many components in the sample and all of them are being that well separated. So I guess it's a good thing that we have all of these components separated and identified. However, if we have to identify those, that's the next step, but I would like to explain a little bit later. But right now, just let's take a quick overview. Why is it so fast? And it's, as a matter of fact, up to 5 times faster than the HILIC methodology. So the sample preparation in an hour. With UPLC methodology, you can have these with all the included steps what I mentioned. Not like 5 minutes is the sample, the labeling or something. No. You have to use your pipette, or you have to actually put your vials actually in the centrifuge and put your vials in the heating block and so forth. So it takes up to 1 to 3 hours practically. The separation with HILIC, even though you can use short columns, however, if you would like to get the same separation power and regenerating the column, it takes up to 45 minutes. In our conventional capillary electrophoresis unit and capillary electrophoresis kit, that had been on the market for like 2 decades, the sample preparation was like at least 4 hours and the separation time was 20 minutes. With this new fast glycan separation, labeling and separation kit, we have the sample preparation done in 1 hour and the separation itself is in 5 minutes. So it's very cost effective. It gives you immediate glycan identification with the structures, and it's a very cost-effective technology. So the kit itself, as I mentioned, won the Best Technology Application in Analytical Chemistry Award last year in the Bioprocess International Symposium. And the kit itself contains all the components which are necessary for the work, even including a pre-built capillary cartridge, which actually really eases up the separation part. And it has the proprietary separation gel that is capable of providing very high resolution separation, a small magnetic separator which can be used for like a couple of samples. Obviously, if somebody has dozens of samples or like 10, 20, something, then it's better to use like a 96 well magnetic plate. It has all the sample processing buffers, the labeling dye, the bracketing standards which are necessary for the Glycoinformatics part, and even a glucose ladder standard to check the performance of the kit and the performance of the separation system. So I have been mentioning Glycoinformatics a couple of times because that's one of the key components of this new kit as well. And that is based on the glucose unit calculation, which glucose units can be used for searching databases, identifying the structures without getting into like additional exoglycosides, digestion steps, and so forth. So the glucose unit can be calculated stepwise like a iteration processes like the Kovacs indexing gas chromatography, or with a polynomial fashion. However, and the significance of the glucose values are it's an abstract unit to express the migration time. It's a normalized migration time in that extent, and it's instrument independent. It predicts actually even exoglycosidase digest chips, and the databases can be used to retrieve the information. How does it work? Original idea is to run the ladder, which is in the upper trace right here, and then run the sample. Based on that one can just do the calculation based on the polynomial or the just a simple calculation way of the numbers, which can be seen right here, which are the glucose unit values of these major components of this separation. Then what happens? It goes into the database, and the database provides the structures which are corresponding to those glucose unit values. This is the old-fashioned way to do it, requiring running the ladder and running your sample. And if you would like to be very, very precise, you run the sample, the ladder again, and the other sample. So it always doubles up the separation time requirement. But actually, SCIEX came up with the help of University of Debrecen colleagues, that we have this patent-pending technology of using only 3 of these glucose ladder components, the maltose, the maltotriose, and the maltopentadecaose, the 2, the 3, and the 15 MERS. Because with that one, using the original ladder, we can calculate a virtual ladder, which would be good enough to define the glucose unit values. And in this case, these 3 components, again, the maltose, the maltotriose, and the 15-mer are co-injected with each of the samples. As one can see, this is the separation right down here of a regular IgG molecule. The maltose and the maltotriose are migrating very much faster than any components of that, and the 15-mer is migrating very much later, so it's practically brackets everything. However, the separation temperature is key to really get those numbers correct. As one can see here, running the same sample, the same oligosaccharide mixture of the maltose oligosaccharide mixture and the IgG glycans at 20 degrees centigrade, the peak number 1 has around like a degree of polymerization close to 8, so the glucose unit value close to 8. If we do the same thing at 50 degrees centigrade, which is 30 degrees centigrade higher, I understand, but as one can see, it migrates close to the 7 MER. So it's a 1 glucose unit value change. And of course, you can't expect a 30 degrees centigrade increase with any of the systems, but a couple of degrees of centigrade you definitely have. And if you actually use the Arrhenius equation to understand the activation energy requirement of these different structures for the electrophoretic movement, what happens is for the linear or helical structure, malto-oligosaccharides, and the branch structures, the activation energy requirement is completely different. You can see from these 2 lines that even a couple of degrees of centigrade changes, if you can't control it well, will actually shift the glucose unit value, which means that you call the wrong components. So this new triple internal standard-based structural assignment methodology was developed to really fulfill this function. And the suggested approach alleviates the need of any accompanying malto-oligosaccharide run, the ladder run, for the glucose unit calculation, which significantly speeding up the structural assignment and processing time. This slide shows a typical separation after the sample preparation and injection on the P800 plus capillary electrophoresis system with the bracketing standards, the DP2, DP3, and DP15 right here, and of course all the components which were released from the monoclonal antibody molecule, accompanied by a table that shows all the migration times, the names of the molecules which are coded from the internal database, and their glucose unit values. And this comprehensive glucose unit database is built in the 32 karat software version of the software of the P800 Plus instrument, and it's again compatible with the 32 karat version 10 software. Is downloaded, it will be downloadable by the release of the kit from the SCIEX website, and it has the added ability to have additional glycan structures to this database. So if somebody is using the original database downloaded from the SCIEX website, can add additional structures to it for their own components. So it will contain actually initially just the IgG glycan main types. It's like 30, 40, 39 as a matter of fact glycans, but if somebody has like other glycans, can easily add to that database and do the search accordingly. The other main advantage, as I mentioned earlier during the explanation of how the sample preparation works, is the automation function. So it can be fully automated because the entire process has been designed and implemented in such a way that a liquid handling robot can be readily used for sample preparation. Again, the single-use magnetic manifold which is provided with the kit can be used for a couple of samples. Of course, it's not very convenient if you have dozens of samples. If you have dozens of samples, it's maybe better to use a liquid handling robot, and we, as a matter of fact, at SCIEX have tried and evaluated with the Biomac robot, and it works just beautifully. And up to 96 samples, one can do the sample preparation, again, within 60 minutes. And this is just such raw data shown here using the sample preparation and the robotic system, the Biomac FX robot, and checking the separation of those 96 samples for the major components of the glycans. And we got an RSD of the GU value less than 1.07% precision of the calculation and the calling of these individual structures. So the features of this fast glycan labeling and analysis kit are the following. It's utilizing a novel magnetic bead mediated sample preparation protocol. Up to 96 samples can be processed in 60 minutes. In a microtiter plate format, we do the requirement of centrifugation or any vacuum centrifugation step, which is very important. For users analyzing only a small number of samples, the single tube manual magnetic manifold is provided. The ultra-fast analysis was worked out, which resulted in very, very rapid separation of the label samples. And the protocol was also implemented using a Biomac laboratory automation workstation if somebody has really dozens or even up to 96 samples. So if we revisit the original industry challenges, what can we see here? Unfortunately, today we have lengthy separation preparation. No, not anymore. You can replace it very easily with the 60-minute fully automatable process what we introduced. I have long separations, not anymore. The 5-minute separation provided by a kit and the Capillary Electrophoresis unit is really giving us a beautiful separation, very, very high resolution. Complexity of the analysis is not complex anymore because we have preloaded methodologies. It's just like hit start and it does it. And data analysis and identification, again, is fully automated. It's built in the software but is provided with the kit. So the fast labeling, fast glycan labeling and analysis kit itself, again, it really works up to 5 times faster than HILIC. The sample preparation is in 1 hour. The separation itself is 5 minutes, and it's automated identification in a very, very short time period. Again, the kit itself provides all components what's necessary for the work, in addition to a pre-built cartridge. And the pre-built cartridge has benefits as well because it gives us uniform, reliable injections with the precision cut tips of the end of the capillary, eliminating actually any kind of injection problem and run-to-run variations. It improves robustness and reproducibility, And of course, it saves time and increases productivity because nobody has to really build this cartridge for the work itself. We have a technical note on the fast glycan labeling and analysis, which can be downloadable from the SCIEX website. And we have a high-impact publication in analytical chemistry or novel patent-pending triple internal standard-based glycan structural assignment methodology. So if we would like to check the components of the fast glycan technology, of course the centerpiece is the kit itself, which is containing the pre-assembled capillary cartridge being used for the P800 Plus pharmaceutical analysis system. However, the CSI-8000 Plus platform, which actually works with mass spectrometry, can be used as well because it can accommodate fluorescent detection. And finally, a liquid handling robot can be very, very useful if someone has not only a couple of samples, but dozens of samples. With that one, I just would like to acknowledge my coworkers at SCIEX, my boss Jeff Chapman, and Mark, Clarence, Marsha, Murph, Anna, and Eric, who actually did a lot of work to be sure that this kit is delivering what we dreamed about in the first place. And my former student Csaba Váradi, who is working at NIBIRT right now, and Martin Szigeti, who worked on the details of the kit itself, and Gábor Járvás, who helped a lot with the software implementation. And with that one, I just would like to actually go to the next question and answer session, and thank you very much for your kind attention.
Thank you, Dr. Guttman, for that informative presentation. Before we begin the Q&A session, I would like to advise that a poll question will appear on your screen. So you may select your answers, then close the poll question by clicking on the X in the right corner. So it is time for Q&A. If you have a question you'd like to ask Dr. Guttman, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is your peptide N-glycosylase F digestion effective on IgG Fab-linked glycan?
As a matter of fact, it is what we have been using for the development of the kit. So it is, yes.
How about the in-between samples capillary equilibration time?
We don't need any capillary equilibration time between the rounds because we replace the buffer system itself, and it's the beer silica capillary with a low pH buffer, so we don't need any equilibration time. We can just run straight right after each other the sample.
Do you use a coated capillary in the CE separation?
No.
Okay. And classically, glucose unit values for HPLC have been measured against a dextran ladder, not a malto-oligosaccharide ladder. Why did you change?
In this extent, it's completely irrelevant which ladder is being used. We changed that 20 years ago when the first kit was released by Beckman Instrument using the malto-oligosaccharide ladder. And we just wanted to keep actually the same ladder for this kit as well because people have been using it and people have been satisfied with the use of that ladder. On the other hand, as I mentioned, we don't even use the ladder, as a matter of fact, because we only use the bracketing standards and the internal standard, the maltose, the maltotriose, and the pentodecaose, and building up the virtual ladder, not the real one, to save time of the separation.
So an attendee says something is not yet available on the SCIEX website. When will it be released?
Excuse me, can you repeat the question?
Yes. Something that you're talking about is not yet available on the SCIEX website. When will it be released?
That's a marketing-related question. I am on the application side. So whenever the kit will be available for purchase, it will be available from the website.
Great. What other assays can I run on the same instrument?
On the CE instrument, not provided like the P800 Plus and even the CSI-8000 systems, all other kinds of like SCIEX kits like the CSDS, the CE-LIF, and all other kinds of works can be run on those systems. On the CSI 8000, of course, it can be connected with mass spectrometry as well, which is a very important feature.
What lab equipment do I need for the labeling and cleanup steps?
Oh, not too much. Of course, the pipettors, the heating block, which should be precisely set, and the vortex mixer, that's it.
Can this approach be used with mass spectrometry detection?
Yes, it can be used, and we are working on it as of now, and try to put together an application note on the use of CSI-MS with APTS labeled glycan. Yes.
Can your method differentiate between 2,3 and 2,6 linked sialic acid?
Yes, it can actually. We proved it with the separation of fetching glycan.
What is the dilution factor of the sample prep process? For instance, the ratio of initial concentration in the original sample versus the concentration of the glycans in the processed sample.
Factor is it depends on the glycosylation extent of the glycoprotein itself. So it's not like there is no straight answer for that one, because the IgG molecule itself, for example, contains 2% glycosylation. The EPO contains 40% glycosylation, so it means that you have to start with a completely different amount of those samples to be sure that you have enough glycans. So from EPO, you can start with a couple of micrograms. From IgG, you have to use at least like up to 25, 50 micrograms to be sure that you see at the end all the glycans with a very, very small glycans with like in an extent of like 0.1% as well. So it's really dependent on the sample itself.
What are the losses of the sample during the sample prep?
What are the losses during the sample prep? When we originally worked out the sample preparation, we used actually radioisotopically labeled sugar molecules, and we did determine that it was better than 90%. So we maximum losing 10%.
Can you give me the name and brand of the magnetic beads?
The magnetic beads are proprietary for our company, and it's actually part of the kit.
Thank you. What about more complex triantennary or tetraantennary glycan analysis?
At this stage, actually, we were focusing on the biantennary structures of the IgG molecules because, as I shown in one of my first slides, that is what the majority of the industry is interested in. The next step will be going forward with tri- and tetraantennary structures and including them in the database as well.
How many samples can you analyze with one kit?
Up to 100 samples.
It seems separation gel is included in the kit, so it is actually CGE, capillary gel electrophoresis, not CE?
It is what I would consider that, yes, that's correct. It's definitely, I would consider this one as capillary gel electrophoresis, yes.
Do you need to analyze purified samples, and what is the lower limit of protein that can be analyzed?
It's the same answer as I had before. It really depends on the glycosylation extent of the glycoprotein itself. If it's very heavily glycosylated, it's really like the low, even sub-microgram range. If it's very, very lightly glycosylated, it has to start from like a couple of dozen micrograms. Again, it's sample dependent. There is no straight answer for that.
Okay, and I can see that more questions are coming in, so I think we will pause for a moment to let the questions settle in. And let's see. Let's just give it a moment.
Sure.
You know, I'm just thinking while we're waiting for the additional questions to filter through, I'm just wondering if you have any final thoughts. I realize more questions could come in and it could duplicate, but if you have any final comments or thoughts about your talk today.
Again, the thing what I kept on emphasizing, and I think is the major take-home message, is that this 60-minute sample preparation includes all the hands-on steps. So if you start and finish in 60 minutes, it's done. The other important thing is that it doesn't require any centrifugation or vacuum centrifugation steps, which eases up the automation option. Because if you would like to have a full automation and you have a centrifugation step, that really increases the price of the robot what you should use. With the system what we worked out here, it doesn't require that, so simple liquid handling systems can be used for full automation of the sample preparation, which can be done again within 60 minutes. And after that, a very rapid separation of 5-minute separation time, one can get the answer immediately. And again, if it's like the bioprocessing question, then getting the sample from the processor, 1 hour later, 1 hour and 5 minutes later practically, have the answer.
Okay, well, I would like to once again thank you, Dr. Guttman, for your presentation. And I would also like to thank our sponsor, SCIEX, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September 24th, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.
Thank you.